The point of today's presentation is to give everyone some information about the training modules being offered through LSU's Gene Lab this semester. Their focus is always going to be on data science and bioinformatics. It's offered primarily through the OmicsLogic portal, which is offered by Pine Biotech, as well as their backing from the Tober Institution. So just to give you a brief overview, there are 3 different training modules that are going to be offered this semester. Each semester there'll be different modules being offered, but there's just 3 for now. The big push for these training modules is that they're completely asynchronous. So you guys can take them at whatever rate is best for you. So if you take longer to learn some subjects, you can take your time to go through those subjects, or if you're quicker in others, you can move faster. But you're not going to be doing this entirely alone. Like, these programs are intended to teach you how to do things properly, right? So there are going to also be mentor-led lectures, which are also asynchronous. So if your schedule doesn't line up with the lecture times, or you aren't available one day over another, you can always watch the lectures at a later date because they'll be entirely recorded. There's also going to be a variety of tools that are available to help you through the programs. So one of them is going to be the online chat assistance. So you guys can always ask questions through a variety of tools that you'll discuss in each individual program's orientation. So you'll always have experts available to you if you get stuck, or if you have a more advanced question that you don't know is actually if it's covered in the training module or not, there are experts available at all times. If you're with LSU or coming through LBRN and you have an indirect question, or you don't know who to ask the question to, you'll probably be directing that question to me. My name is Lane Yutzi. Some of you may know me through the PBS department at LSU School of Vet Med. But if you need to reach me at any time, we will have the information in the chat. My email is lyutzy1 at lsu.edu, and I'll put that in the chat right now for you. So if you have any indirect questions that aren't answered today, or if you don't think of them at this moment, you can always reach out to me at that email address. That being said, once you complete any aspect of this program or the entirety of the program, you'll receive a certificate of completion. And this is going to be different. I'm going to show you some of this information later on, but this is going to be a unique certificate to whatever training module that you take. And it'll be signed and backed by an individual of your institution. So if you're with LSU, you're going to have that certificate signed by Dr. Gus Kasulis. And if you're with LBRN, the person is tentative, but it is likely to be Chris Taylor, if you're familiar with him. So how are you going to take these training modules? How are they going to be delivered to you? There are 3 primary platforms that we're going to be discussing throughout this lecture here. The biggest one you're going to interact with is the OmicsLogic portal. This is essentially a website that gives you access to all of the lessons, all of the program information, and all of the assignments that you'll utilize throughout this training module. I'll show you a bit more in detail later on how this portal works and what it looks like, but this is the primary one you'll be interacting with. If you're taking one of the more advanced courses, you'll also have experience with their T-Bio platform. And essentially what this is it's a flowchart system in which you can take whatever data set you're going to be utilizing, and it's going to work you step by step through the process to analyze that data and visualize that data for whatever desired outcome you have. But this is going to be more for individuals that are going through the advanced projects. Also, another tool that will be available to you that is really important for this process is the Python and R coding playground. So for those of you that aren't as familiar with coding, like myself, as I was originally, whenever you code something, you need to create a proper coding environment for the coding to actually run properly. Now that can be kind of difficult, especially so I had no experience prior to working with this platform. So I didn't know what I was doing, but luckily the OmicsLogic platform provides a coding playground for you. So you don't have to do any of the setup. You can just practice the coding steps yourself within their coding playground. So it makes it a lot easier for you to understand these processes. And so there's 2 links that were just put in the chat. That's some more information to the OmicsLogic portal, as well as the T-Bio platform if you're curious. So there's 3 different difficulty levels that I'm going to be discussing with these training modules: beginner, intermediate, and advanced. So to give you an idea of where you may fall in those three categories, I'm going to describe a little bit about what we consider a beginner, an intermediate, and an advanced level of expertise. So a beginner to us is someone who has minimal background, both biological or data science. You have maybe heard of bioinformatics, or you're familiar with the concept of it, and you're curious to learn more about it, how it's used in the industrial, research, or clinical worlds, then you'd be considered more of a beginner. If you're an intermediate level, that would mean you'd have some kind of understanding of biological and data science concepts. Maybe you have a little bit of background in either one. You've had a little bit of statistical experience. Maybe you've taken a statistics course, or you are familiar with a little bit of the concepts, and you have maybe some coding experience. An advanced individual is someone who has full understanding of the verbiage used in biological and computational fields. So you understand whatever you're reading. You're not really hung up on a lot of the words, right? You have some statistical experience. Again, maybe you've taken a course or two, you've applied some stats to your own projects. And then you have some coding experience with R or Python. So if you have statistical experience, most likely you have heard of or worked with R to some degree. And you also understand next generation sequencing to some extent. If you have a biological background, which I'm sure many of you do, you've probably learned how next generation sequencing works, or at least at the very minimal heard about the different types of next generation sequencing that are growing in the biotechnical field. So let's talk about each individual training module that's being offered this semester and what each module brings to the table, right? So the first module I'll be discussing is the beginner level, getting started with bioinformatics. You'll also hear this referred to as Introduction to Big Data Bioinformatics or Introduction to Bioinformatics, but it's the same thing across the board. This is, like I said, a beginner level training module with a rough time commitment equivalent to about a 1 credit course. If we're thinking about an equivalent to the difficulty level, there's probably more of a 100 or 1000 level, depending on what institution you're from. So it's very introductory. It's just to give you an overview of the key topics and the applications of bioinformatics. That's from everything from the basics of the biology that you would need to understand this information, as well as applications of big data in the modern world, right? So if you don't have any background, or you only know a little bit and you just kind of want to dip your toe in, this might be the training module you're interested in. Now, our intermediate training module that's being offered this semester is data science for biomedical data. This is more equivalent to a 2 credit hour course and is closer to maybe a 200 or 300 level in the academic world. The purpose of this training module is to teach you methods for how to wrangle data, analyze, visualize all of your data, as well as a little bit about how machine learning is applied to bioinformatic data. So all of these concepts that you'll be discussing and practicing are actually used in the analysis of omics in the real world. So it's to kind of give you some of the tools you would need to understand and even perform some of the data analysis that's used in omics studies. The third training module that's being offered in the advanced field is the bulk and single cell transcriptomics training module. Like I said, this is advanced. So this is someone who already knows that they want to learn specifically about transcriptomics, but maybe you want to learn more about it or how to perform the steps of analysis yourself. So that's the purpose of this training module, is to help you learn some of the methods as well as visualization techniques that are used to analyze both bulk and single cell RNA-seq. So if you're thinking about a way to integrate data analysis, specifically RNA-seq data analysis, into your own work, this would be a great training module for you. So let's get into a little more nitty-gritty of each individual training module as to what are the subjects covered, et cetera, right? So first things first, each training module is going to have their own individual orientation that will give you real in-depth information about which tools you'll need to access, your expert help, when lectures are going to be available, and things like that. However, even though the orientation for getting started with bioinformatics is going to be on October 6th, because this is asynchronous, you'll have full access to the OmicsLogic portal on October 4th. So if you want to get a head start and take some of the courses or some of the lessons that are available to you in this training module, you'll be able to do so at your own rate. So what exactly is getting started with bioinformatics. And what will it cover? So this is basically an introduction to everything bioinformatics and data science. It's to give you a strong foundation and the understanding of how to apply this information to your own work, or sorry, how to apply this information in the world, in the real world. And so it's going to cover a variety of concepts to give you an idea of what exactly is out there in the bioinformatics world. So first, we're going to start with concepts. You'll also get introductions to some of the more subspecialties in the omics field, such as genomics or transcriptomics. You'll learn about differential gene expression, machine learning, and how those are applied in the bioinformatic world to help better our understanding of the data we're analyzing. Now, you're going to understand that you're going to learn how this information is applied, not just in the research world, also in the clinical world and the biotechnical world. How can these be applied to different fields throughout specifically the United States, as well as academic institutions and industry around the world, right? You're also going to see hands-on how this is done through a variety of case study-based projects. So you're not just going to talk about conceptually how these things are applied. You're going to study exactly how they have been applied to different specialties around the world, right? So if you're curious and you want to get more information, you can go to the link down below. If it's a little bit a longer link, but if you have access to the informational pamphlets, the link is available there as well. And they'll probably put it in a chat here, so you can just click on the link directly. So these training modules are built off of asynchronous courses. So this particular training module, Getting Started with Bioinformatics, was built primarily off of or inspired by these two training asynchronous training courses, Introduction to Bioinformatics and Bytes and Molecules. So there's a big difference here, though, between the asynchronous courses and these training modules. So if you go to the OmicsLogic portal at learn.omicslogic.com, you can have access to a variety of the lessons free to everyone right now. You don't have to sign up for anything, and you can give you an idea of what's available to individuals through this program, right? So the difference, though, is when you're taking these individual asynchronous courses, you're not getting a strong understanding as to how everything is interconnected. You're learning the syntax and you're understanding everything that's being displayed to you and instructed to you. But the purpose of these training models is to really tie everything together to show you how all of this information can be synchronously combined to your own work, to future work, to different fields, and how everything is interconnected and can be utilized together to progress either your own research or research you're familiar with, right? So that's the purpose of these training modules. They are inspired by these asynchronous courses, but the purpose is to really show you how everything is interconnected and intermingle it with your own work and the different tools available to you. They're broken into different lectures that will help you work your way through this training module. And again, like I mentioned, these are asynchronous. So if you can't make these particular dates, which these are tentative dates, they're open to changing if people have issues with schedules and stuff. If you aren't able to meet these dates, they're always going to be available recorded to you upon this date after they've been delivered, right? So you can reach out and you can watch them at any time that's convenient to you. You can complete things earlier or later at your earliest convenience, right? Each lecture will focus on particular subspecialties, and so you can see some of them here. I'm not going to go through each one, and each one of these own lecture focuses will have different subjects within them that you can see fully at the link provided in the chat. That will give you through the entire syllabus, what subject is in each lecture, what are some of the focuses of that particular subject. You can see all that on the link in the chat right now. So like I just mentioned and kind of discussed, the purpose of Introduction to Bioinformatics is really to provide you with a variety of skills that you will need to understand the bioinformatics world and understand how it's applied in the real world. So you're going to learn a variety of different things. You're going to learn Python and R coding. You're going to learn a little bit about stats. You're going to learn about machine learning and AI guided analysis, specifically in the context of next generation sequencing, right? Because that's what you're usually more interested in, and how you can apply it to your own direct work. And at the end of it all, you're going to get a certification supported from whatever institution you're from, whether that's LSU or LBRN. So if this is particularly interesting to you, or you just want to kind of dip your toe in, this might be a great training module for you. So moving on, the next training module that we're going to discuss is the data science for biomedical data. The orientation to this particular training module is October 7th, again at 12 p.m. Central Time. And again, your access is fully available from the 4th. You're going to see that across the board. October 4th, you can access all of the training modules that you sign up for. So what is the purpose of this particular training module? The idea here is to show you how big data is mixed with biology. How can it be applied directly to biological studies? And how is it being used already, right? As we further our knowledge about biotechnical research, you've already noticed throughout your research papers that more and more individuals are integrating things like data science into their own studies. It's a great way to further our understanding of our samples, to really see how massive changes are occurring in different cell states, and whatever your studies particularly apply to. I'm sure you've noticed a growing focus on data science integration. So this is what this training module is trying to show you, right? You're going to learn a lot of different things. You're not going through this alone. As a biologist, you don't always have a lot of exposure to data science. Usually it's entirely on the biology side of things, right? So there's a variety of tools that are going to be available to help you through this process. There's hands-on exercises, tutorials, there's coding challenges, which if you're not familiar with coding, it'll help work you through the steps. It'll teach you the functions and things that you need to create to run the processes you're trying to do. As well as you're going to see real research examples. We're all familiar with images like this. We've all seen studies and papers like this, right? So you're finally going to understand and get to learn how people are taking some of their data and they're creating visualizations like this, how they're getting to these research papers. That's what the point of this particular training module is to learn how to analyze that bioinformatic data, all the way from wrangling your data through visualizing your data. So there's a variety of concepts you're going to learn here. This is just a few of them. This isn't all of them. But to give you an idea, you're going to learn about statistical concepts, machine learning and deep learning. You're going to learn some Python and R coding, and general coding practices associated with bioinformatics, as well as data sharing. And you're going to learn these both conceptually and how they're applied practically to the real world for both research and clinical applications. And you're going to get to do it yourself. So this isn't just learning about this. You're going to practice yourself with coding challenges and different assignments along the way. These are training modules. They're not just dumps of information to read, for you to read on your own. You'll have ways to apply this information, to practice this information yourself. And if you're more curious, you can go to the link that was provided in the chat for this particular program. Just like the last one, here is the syllabus, at least the lecture focuses of the syllabus, and some of the individual asynchronous courses that were used in the inspiration of this particular training module. You can see some of the stuff you're going to learn throughout the course in more depth here. Unsupervised learning, specifically clustering, supervised learning, classification. If this is interesting to you, or you could see how well you could apply biomedical data analysis to your own work, this might be the training module for you. This is the intermediate training module, and like I mentioned before, it is offered orientation for this training module is October 7th. So our advanced training module being offered this semester is the bulk and single cell transcriptomics training module. The orientation for this one is October 8th. So you can, if you've noticed the trend, I'm sure, introduction is on the 6th, biomedical data is on the 7th, and then transcriptomics is on the 8th. Again, all access is available October 4th. So if you want to get a head start, you're more than welcome to. So what's the point of this particular training module? It's to provide you with a variety of tools on how transcriptomics is directly applied to your own research or research that you wish to get into in the future. You're going to learn a variety of methods to help you do this, different analysis methods, statistical analysis, as well as clustering information, data mining, classification of data. And so eventually you'll be able to visualize whatever data you're working with in a variety of ways. So this is for individuals that really know that they want to integrate transcriptomics into their own work. And And fortunately for you, if you have your own data already and you're just curious as to how to analyze it and apply it, that is, you're able to actually do that in this training module. You're going to learn about how to perform transcriptomic related data analysis and visualize that data. But you're also going to have a project later on that'll take you from start to finish of a whole transcriptomic project. So if you have your own data, you can actually use that there and do the project with your own data. So you're progressing your own research potentially while learning about how to do these transcriptomic data analysis, right? You're going to learn about a lot of different things in this module, and this is why it's considered more of an advanced module. You'll understand more in-depth concepts with machine learning specifically for expression-based data, exploratory data analysis. You're going to learn about how to analyze and process gene expression data, specifically that associated with next-generation sequencing. You're going to learn how to data mine, as well as you're going to learn how to apply this through the biological interpretations that you may have heard. So I'm sure many of you are familiar with pathway analysis or have seen papers discussing pathway analysis. You're going to learn how to do that here with your own data. So this particular training module is for individuals that have some background in all of the information discussed and want to really further that and integrate it into their own transcriptomic studies, right? That can also be bulk and single cell. So if you have one particular one in mind, you'll learn about both in this training module. So here's the syllabus, and again, some of the asynchronous courses that have inspired this training module. I'm not going to go through in full detail, but again, the orientation is on October 8th. And you can see some of the subjects discussed in this particular training module. To give you an idea of all of the things that are going to be there to help you through this process, this slide gives you a lot of information as to how this is really designed to help you succeed. It's not just leaving you high and dry to learn the information on your own. There's a variety of courses and tutorials available to you. As I mentioned, there are multiple mentors that will be available to help you through the process, right? You're going to be guiding yourself, but there's plenty of tools that are there to make sure that you succeed. All the methods that are going to be discussed will be explained to you so you can fully understand why you're doing what you're doing. And you can start to understand different ways to help improve the analysis that you're doing with multi-omics AI guided analysis. You start to understand gene expression data better, as well as data science. You'll do some of the coding, like I mentioned. And then you'll see how this is directly applied to your own fields of expertise, right? Your own biomedical next generation sequencing research could be furthered from this training module, as well as that project that I mentioned has the potential to eventually be, you could present it as a proposal or a poster or presentation at a conference in the future. So speaking of that particular potential, there are some available resources too that you may not be aware of. So if you guys are from LSU or LBRN, there is the LSU Gene Lab available to you. Gene Lab provides a variety of consulting and experimental services that could be beneficial to your research. So Gene Lab, some of the services, this is just some, if you're curious, you can go to their website to learn more, but they offer a variety of sequencing and next generation sequencing services, PCR services, gel imaging, multiplex protein analysis, and as well as automated cell counting services at Gene Lab. If you're with LBRN, there's the LBRN Bioinformatics, Biostatistics, and Computational Biology Core, which is designed to provide a consulting, mentorship, and training through their bioinformatics liaisons. This is primarily focused towards PIs, but so you can reach out to them directly to learn more about how to be furtherly trained in these fields. As I mentioned about that project, the project that you could present, there is the annual LBRN conference on computational biology and bioinformatics. This is usually in the spring. Last year, it was around April. So you have the potential to present your projects that you're working on at this conference or a similar conference of your choice, if you choose to do so. There are always ways to continue furthering your education. Now, if you're a faculty member here today and you're curious about these programs, there are a variety of other training modules offered through Pine Biotech that if you are curious about how to offer programs like this to your own institution or to your own students, You can reach out to Ilya Brodsky. He is in the call today. He's the CEO of Pine Biotech. His email is ilya@pine.bio. So if you email him, you can get a variety of information about some of the programs and training modules that have been offered at a variety of institutions across the country, as well as throughout Louisiana. So if that's something that may interest you, definitely reach out to Ilya. So if anyone is interested, I'm going to reiterate one more time that all access begins October 4th. What I haven't mentioned is that all access will end December 31st. So if you have a busy schedule, or you really have to structure your schedule, or you're worried about, well, these courses may overlap with some of my own courses at my academic institution, that's fine. You have plenty of time to complete all of the information as long as you keep up with it, right? It has to be treated like a normal one of your courses, but you'll have plenty of time to complete assignments and balance your schedule accordingly. This shouldn't be overwhelming to you. No one wants it to be overwhelming to you. So the best thing, that's why the training models will last until December 31st. So if you need additional time, you can schedule accordingly. So the next thing I'm going to do is I'm going to show everyone how to register on both the Learn OmicsLogic portal, as well as how to register for these courses. So we'll now open up to questions before I do that. And I see one question in the chat: can you enroll in more than one of these courses? You can at the moment, but it has to be approved. So the issue is we don't want someone with a background that say you want to take intro and you want to take transcriptomics, right? So those are different difficulty levels. We don't want one individual to go into transcriptomics and not have the background they need to excel, right? So it is possible, but it has to be approved from the organization that you're associated with. Is it possible to only audit the courses?
Yeah, no, I think after this, you're going to be showing the portal, and on that portal, in each course, there are some free lessons. So if you just register, take a look at some of those examples, and then decide if that's something that you want to do, and proceed with the full registration.
Okay, so if you guys go to learn.omiclogics.com, which it has been mentioned in the chat, there it is again. So if you guys go here, you'll see clearly in the center of the page, register for free. So if you want to register, like I said, there are some training lessons available for free. You don't have to register for anything in terms of these programs. They're available to everyone. So if you want to get an idea of how the portal is structured, that's a great way to do so. So you're going to click here for register for free. And so if you don't already have an account, you're going to click right here to create an account. All you have to do is put your information in. Your name, email, and make a password, right? Or you can also sign up with a variety of other methods: Google, Facebook, Apple, etc. Okay, and it'll take you here. So the biggest thing that you're going to want to remember to do is go to your profile. That's the easiest way to access everything. So once you get to your profile, this is what it'll look like. You might not have a profile picture, or a description, but feel free to put that information in. You can also link your socials if you choose to do so, like Indeed. And you'll notice right away, you'll have a subscription down here and a level. So as you complete different lessons and go through the program, this level will increase. So you get points for completing things. You can see here, I got 500 for completing, starting with Bioinformatics and Python. And your goal is eventually get to 10,000. The reason the goal is to get to 10,000 is once you get to 10,000, you'll unlock projects, which if you follow the program appropriately, you'll get 10,000 before you're required to start your project. Now, when you first make your account, you'll notice your subscription is probably different, and you won't have any badges down here. But once you register for a training module, the training module you register for will come up down here as a badge. So you can see here's the different training modules that we discussed today, right? So the point of this is, once you start a course or a project or a program, you can see all of them here at the top. If you go to courses, you'll see all the courses that you've started working on. So as you can see, I've completed course 1 here, Python course 1. And as you work through it, you'll start to see that this value increases as you complete the different lessons within the courses. Now, once you complete a course, you'll still get a certificate for completing that course, similar to the one that you'll get at the end for completing the program. But the one you'll get at the end when you complete your program is specific to that program and to the institution. So you can use that as an indication that you've been trained in bioinformatics in that particular subject. So what do these certificates look like? If this loads, I can give you an idea of what it looks like. Okay, so this is kind of what your certificate would look like, except your name will be here. This is just for the lesson, right? So as I mentioned, it's going to be different for once you complete your program. But once you complete the program, you'll probably see decals for LSU or LBRN, depending on what institution you're working with. And it'll also be signed by a specific individual. So Dr. Augusta Solis will sign here if you're LSU or potentially someone from LBRN, as well as the date as to when you finish the program, right? So going back, if you want to find out the programs you're in, you can go to the programs tag here or tab here, and this will show you what your progress is in the program. It'll also give you information about the program, right? So if you sign up for BioMed Introduction to Bioinformatics or Data Science or Transcriptomics, like we just discussed, this will show up here as well. So if you want to find it, you may not see it down here, you can go to the Programs tab. If you're starting new and you want to find new courses, you can go to this course selection here and search different introductory courses at your leisure. If you get to the project, so once you get to 10,000 points in the project, this will unlock and it'll take you step by step through how to make a project on OmicsLogic from start to finish, from choosing your data set, choosing your hypothesis, and working through the different steps associated with the project. So that being said, the next thing I want to show you how to do, this is how to register on OmicsLogic. If you want to register for the training modules, you're going to go to iLabs, which is at ilabsolutions.com/account/login. So if you don't have an iLab account, you're going to have to make an iLab account. I'll wait. If you want to do this with me and you already have an iLab account, I'll go ahead and hold up for a second. But for those individuals that don't, I will also put a document in the chat that will take you step by step how to do it. So you'll have that available to you if you choose to register. So that document will help you go through it step by step if you miss a step today while I'm talking about it and still want to register, or if you haven't made a decision yet and you just want to sit on it for a little bit, just download that document and you'll be able to register at any time. So actually, we're going to log in with another individual's account who has fortunately given us their access information to kind of show you what it's going to look like. Since I'm an admin on the account, it looks a little different for me. So once you sign in here, the first thing you're going to notice is this screen. You're going to log in. You should see something like this. You might not see the service request down here, but you'll see the home page. Okay. What you're going to want to do is you're going to go up here to this drop-down menu in the top left. When you click on that drop-down menu, you're going to want to look for Core Facilities. Okay. If you click on Core Facilities, it'll take you to the variety of core facilities offered through LSU or the LBRN network. The one you're going to want to look for is the CEDAR Core Facilities and Services, the CEIDR Core Facilities and Services. Primary contact is Thea. You're going to click on that. Okay. Oops. So I have to stop sharing this for a second because of some personal information that should not be shown. Actually, so let me show you first. So first, you're going to want to click, once you get to this page, you're going to want to click on Request Services up here in the View My Requests, okay? Click on Request Services, and that'll take you to a new page. When you get to the request services page, most of you should have this drop down. If not, go ahead and click the drop down box for project requests. And then you're going to want to look for training modules. Here are the 3 training modules that we discussed today. You can see you're going to want to click on request service here on the right side. Once you've found training modules, select request service. So like I mentioned, I'm going to stop sharing just for a second because I need to hide some information. Okay, but once you get there, you'll have a portion at the top will have your identification information, like your lab, your phone number, your email, right? Scroll down just a little bit until you find training modules, and it should have a little red star next to it. You'll see here that these are the 3 training modules that we discussed today. In the training module box, type whichever one of those training modules you want to take part in. If you want to take part in more than one or are curious if you're able to, type that in there as well, and I'll be in contact with you through iLab. Once you've done that, and it's a little easier if you don't want to type it, you can just highlight and copy and paste. Once you've done that, scroll down a little bit farther until you see payment information. Once you've found payment information, there'll be a small box that says fund number. This is not a required field, but to help us figure out which institution you're associated with, type in this box whether you're with LSU or whether you're with LBRN. Once you have done that, all you have to do is click here, submit request to court, and you're done. So once you do that, your registration is complete. You may get one additional email from iLab saying something about costs. Don't worry about that. That's just for our purposes. It informs you that we are processing your information. Now, if you're stuck on this, like I said, download that document. You'll be able to go. It takes you through step by step through all of the information we discussed and how to do it. So, or if you have any additional questions, feel free to throw them in the chat now. Or if you have another question you think of later, like I said, you can always reach out to me at lyutzy1@lsu.edu. So with that, I'll open it up. Oh yes. So I also want to show you some of the landing pages. So the links that everyone linked here in the document as well, to kind of give you an idea of what the landing pages will look like, right? So if you go to one of these landing pages or one of the links provided in the box, you'll see this. So this is your getting started with bioinformatics landing page. This will give you more information about that particular training module if you're curious or want to reread some of the stuff that we discussed today. Go to one of these links, and this is how I'll present to you. So you'll have a brief description of what this particular training module is intended to show you. It'll tell you when the orientation session is for that individual training session. As I mentioned, it's the 6th for the introduction course, 7th for big data bioinformatics, and then 8th for the transcriptomics training module. You'll have a short video available to you that will kind of give you a bit more detail about what the goals of the training module are and what's included in it, as well as some of the key topics that are going to be covered. And what is really important is if you really want an in-depth overview of how this training module is going to function, you can continue to scroll down and you'll see the full program syllabus for that particular training module. It'll cover all of the major topics as well as some of the minor topics I discussed for each particular lecture. That will be covered. So example, as I mentioned, you're going to get introduction to genomics and transcriptomics for the introductory course. So here's some of the subpoints that are covered in the genomics lecture: targeted sequencing, whole exome sequencing, whole genome sequencing, et cetera, et cetera. You'll also see more information about each one of the subject matters discussed, because I only went over a few of them in this particular lecture. Now, if you, like I mentioned before, if you do need help or you run into any issues or something skips your memory as to what to do, you can always contact me at eliotsu1@lsu.edu, or you can join the orientation session for that particular program, which will have links in the future. Now, if you're curious and you want more information, or you want to show your interest, go ahead and go down to any of those pages and register here, listing your information in these boxes to help us figure out who you're associated with, and then we can reach out to you for more information or how to register if you're with an outside institution. If you're curious as to what some of the asynchronous courses that I discussed, or like a full list of them, here are some of the lists here, as well as this is more close to what the certificate will look like when you complete your training module. Okay. So if you have any more questions or information, go ahead and shoot that in the chat. And otherwise, unless Ilya, if you have anything else you would like to add in, thank you for your time.
Thank you. I know I will just add a couple of points. So the differences between the programs, it sounds like several of the topics are almost the same, but it's really the depth as to each topic that is going to be covered and the specific focus. So even though on all the programs, different types of omics data, different types of methods and categories of methods are going to be touched on, the depth of those topics and the specific focus is going to be very different. For example, in the Introduction to Bioinformatics module, most of the topics are going to be a high-level overview. In the data science, it's going to be primarily downstream analysis, data visualization, and finding patterns using machine learning. In the transcriptomics, it is going to be much more focused on actual biological discovery using those methods of analysis, including both bulk and single-cell RNA-seq data. And it is really intended to kind of help you work with an existing data set. So we'll focus on different types of data that you can either identify in the public domain, or if you have data of your own, you can use that as well. So just wanted to make sure that that is clear.